Good evening, everybody, and welcome to Tuesday Talks. I'm Seema Kumar. I'm the CEO of Cure, and we are so honored today to have somebody who's a big luminary, Christopher Mason. He is the author of The Next 500 Years, which I'm eager to learn more about. You're an entrepreneur, you're a biologist, professor of genomics, computational biology, all of that at Weill Cornell Medicine. And you are trying to save humanity. Thank God for you. And, and humanity not just in the small blue dot, but throughout, right? Interplanetary, through your work at the Mason Lab here in New York City. And you're not only one of the notable scientists here in New York, but you are a leading member of the New York community. And you've been able to spin out 10 companies from your lab, right? And I'm sure there's more coming. And so you've looked at the microbiome, you've looked at metagenomics, the application of computational techniques to analyze large-scale biological data. And big data is that's what it's all about. And your work has actually implications for understanding human health and the role of microorganisms and various different biological processes, right? Some call you a space scientist, others call you a mad scientist. You described yourself as a weirdo, and so you've conducted more than 10 years of research in collaboration with NASA and SpaceX, and focusing on the understanding of space travel and its effects on the human body, particularly at the genomic level, right? Yes. And this includes studying how extended space flights can change you, and the exposure to space will impact the microbiome, gene expression, and other biological processes in the body. And one of the notable projects that you worked on was the NASA Twins study, right, where you analyzed the genetic and molecular changes in astronaut Scott Kelly, who spent nearly a year abroad, the International Space Station, and then you compared it to his identical twin brother, Mark Kelly, who was then a US Senator, and who was on Earth, obviously. And it aimed to uncover how space influences various aspects of the human body. And in 2016, you launched the Global City Sampling Day, which spanned 6 continents, 32 countries, 400 people who helped collect 12,000 samples of DNA, RNA, microbes from the surfaces of subways, buses, airports, hospitals, well-traveled meeting spaces. And where were you when COVID happened? I'll come back to that. The project is, you're still swabbing, right? And I don't know, I met a few citizen scientists earlier this year at Davos who seemed to be citizens who were doing something similar, right? And so anybody here in the audience who has something genetically interesting or mysterious, you may end up swabbing them later.
I carry swabs with me everywhere I go.
Yeah.
Just in case,
Well, thank you so much for being here. Looking forward to the conversation.
Pleasure.
Really, really looking forward to it. So I want to talk to you a little bit about your work. But before that, you are a leading and outstanding member of this community in New York City. So talk to us a little bit about what it's been like to be part of the New York community, and also, the early days of the bio community here, and how you ended up establishing 10 different companies out of your lab at Weill Cornell.
Well, it's been, some of it is having a really good ecosystem. So I've been very fortunate, just in general fortunate. I've had really great people in the lab. I think any laboratory and any workplace is only as good as the people in it. And I've been very blessed. I've had extraordinary scientists, and sometimes entrepreneurs, people who have an idea and think, well, let's get this out of the lab and out into the world, make a product out of it. But also some people that are just really curious, that know an idea sometimes takes a year or two to germinate, or sometimes become something that leads to fruition. So some things take time. And so I think giving people space to explore and be creative is one of my main goals for my laboratory. Is how do I get a space where people can tinker and try a new enzyme, try a new experiment. And I often say, if the experiment is less than $1,000, you should just do it and tell me how the results go. If it's more than that, maybe we should make a plan. But generally, if something's just a small tweak, or you want to just explore just do it, and let's talk the next day and how the experiment goes. So encouraging an environment where people can try new things is one facet. But then also, I didn't originally think, I'm going to start a lab at Cornell, and I'm going to make companies. I'm going to have this be a George Church-like fountain of companies, and many people in the audience know George Church, but which I think at one point I was talking to George, and he's like, I started like 30 companies this year, and at first I was like, wow, that's really cool. But then I was like, I think that might be too many companies. So I think about maybe one per year is a reasonable goal. But there is a need for entrepreneurship, for people to push new ideas, to take sometimes a diagnostic that we had then taken to market during COVID, for example, 4 different diagnostic tests, or taking a new microbiome therapy, a new cancer diagnostic. There are many needs in the environment, in the community for better medical care, treatment, cures, as we're here at the Cure. And that often is birthed in academia, but is best, I think, implemented in industry settings. And industry's not perfect either. You can suddenly lose a whole division at Merck or Novartis or even a small company. And it can be brutal, but there is a real efficiency to the market to, let's say, is this working? Is there a market? Who needs this? How do we get it to them? And you don't get that in academia, but you're not supposed to. Academia is supposed to be this floaty space where people are not sure what they're going to do necessarily, or they'll read for 2 days, not sure, and then do an experiment. So I think you need both. I'd say.
Yeah. So speaking of tinkering, I think you've been known to sort of tinker with genetics and genetic codes and mixed genetic codes and all of that, right? And it can be all about curiosity and urging your students to be curious, right? So you can stop at tinkering, but you haven't stopped at tinkering. You've kind of said, okay, from that tinkering has come out diagnostics, therapeutics, maybe cures in cancer, for example, right? Tell us a little bit of how you did that and what kind of examples you can share with us.
Sure. So some of it is very data-driven. So, for example, CAR T therapies. I think many people were excited when some of the first trials came out of UPenn, where we could, it looks like, cure lymphoma, or maybe we could attack leukemias in an entirely new way, where you engineer these T cells to have a chimeric receptor. And so, okay, we're going to not take a T cell as it normally is, but make life as it could be. And so I think that's a recurring theme in the laboratory. It's not just life as it is, but you first have to know what life is before you can think what life can be. And so this applies to immunotherapy as well as to new creatures on Earth, or even de-extincting creatures. I'll talk about that just today. We actually had a visit from the middle school students. We talked about science in the lab, and we had a visitor of Dr. Buckland, who's here. It's basically, what about de-extinction? Can you bring back species that have been long gone? And these were things that were science fiction, CAR T therapy, engineered cell systems, de-extinction. All of that was completely laughable 10 years ago. But so what we've published, though, are ways where if you look at every clinical trial, you look at every expressive marker on T cells, that helps you build chimeric ones that also have logical constructs. So we've published work on making new kinds of T cells. Or one of my favorites is bringing in evolutionary lessons from other creatures, like tardigrades who can survive in space, put that into human cells and see that they can now resist radiation if they go to space, for example, because they're using the resistance mechanisms from a totally different species that's brought into human cells. And I think if you can be agnostic towards where life gives you lessons, like all creatures can give us lessons, like CRISPR, for example, came from looking in salt marshes and bacteria, and now we have therapeutics that are curing beta thalassemia and sickle cell from just people poking around in microbes. Yeah. So all of life, I think, has lessons to teach us, and we're just scratching the surface.
Speaking of de-extinction, I know pretty much every 10-year-old would love to bring back dinosaurs to life.
This came up this morning.
Right?
Yes,
yes. So Jurassic Park, I mean, that's science fiction, but I mean,
is it? Yeah.
Or is it? Is a question, and that just reminds me that I interviewed Bert Vogelstein once, and he said, if you really want to make a big, a big breakthrough, think like a science fiction writer, not as a scientist, and think about the impossible things that you would dream of and then try to make that happen. And then you talked about radiations and creatures that can survive radiation, et cetera. Like what I've heard when growing up is that the only thing that can survive, radiation is a cockroach. And so, right? And so So talk to us about bringing back dinosaurs and maybe, de-extinct dinosaurs and extinct the cockroaches.
Or mosquitoes. There's a
Or
large
mosquitoes, yes.
people who would many people would just do we really need every species of Anopheles?
Yes.
Probably not. But I'm not going to be the first one to eliminate species. We'll start with bringing them back. The biggest problem with dinosaurs in Jurassic Park, I guess there's two big problems with that movie. The first one is it looks like DNA would stay in amber, but of course DNA doesn't last that long. The record for DNA is maybe over 1 million years, is possibly 2, but not 65 million years. DNA just degrades too fast. And so even though that was a cool part of the movie, it has no basis in reality. But you can infer a lot from other species. So again, if you take every species that exists as a template and part of the genetic armamentarium of possibilities, and you can mix and match, which we can do better and better by projects like the Vertebrate Genome Project that's at Rockefeller with Eric Jarvis, or ways to map just known species. We can infer a bit and use tools to say, well, let's bring this back to a chicken, make a chicken a little bit with a longer tail, take away the wings. There's been announcements already to bring back the dodo, the woolly mammoth, the thylacine. There are others in the works. So there'll be, if all goes well, even a de-extinct species next year that we'll hear about. So I think that there's a lot coming. Dinosaurs will be hard, but like you were saying, if you want to aim for the impossible,
why not?
Then it is a question of genome engineering and design, which actually is the best test of our understanding. Can you say, I want to make a creature like this, that has these specific phenotypes? It's maybe the hardest question in genetics, is I want to make this exact kind of cell that appears at this time with this phenotype. If only we understood genome regulation and genetics that well. We don't yet, but we're getting closer. Every day, every year, we're learning more. And so I think
we're learning something new, every day, and who knows what AI could bring to the table, right? And your book, it's called The Next 500 Years, and you're trying to figure out how to re-engineer human beings. I'm going to simplify it so that you can live in a place that's not just Earth, interplanetary, space, et cetera, et cetera. I am just thinking back to some of my favorite series, Star Wars and Star Trek and all of that, and I'm thinking, okay, back to science fiction, right?
Yeah.
You can make this happen. And so I'm sure that in this twin study that you did, you discovered that there are certain things that we need to adapt about our genetics, genomics, in order to survive an environment like space. How do you do that? And then are you creating, quote unquote, designer humans?
So good, or are you playing God as another term?
Oh, you're playing God, yeah.
Some people have said, well, if we're going to be playing God, we might as well get good at it. It's a flippant response to that question. I take a more nuanced approach, is that that you could say the short answer would be yes, that is what it is. If eliminating disease and controlling evolution is playing God, then I think that is what we're doing. But I think that's what we've always been doing, and to some degree, what life forms have been doing when they reorganize matter itself to create their own vitality. When we change ecosystems to divert a river, when we move crops into a state they've never, like the first versions of corn 600 years ago barely made any kernels, and now we have them routinely at Thanksgiving. But this is to some degree what we've always done, but we have a capacity and scale to do it today that is terrifying to some, but I think new technology is always terrifying, and so it has to be done slowly, carefully, with guardrails. And designer humans we're not making yet, but I think the argument I make in my book and the papers is there are certain circumstances where you are ethically bound to intervene, to modify humans, and so it's generally you don't always have to, because you have to have a great example is He Jiankui, who created the embryos that had the CCR5 deletion. He CRISPRed embryos in secret and then released them onto the world, which is probably the worst possible way to do clinical trials or research in general. It's terrifying, so you don't want to do it that way. And even what's worse is the CCR5 deletion, which gives you a native immunity to most HIV strains, is good, but then you have an increased risk of West Nile virus, and there's complicated factors of genetics. And so that's a big problem, but the biggest problem of all is that there was no unmet medical need. What he did there was not only probably unethical and had many problems and technical problems, but there was no real need for what he did. And that came out when he announced it. But there are two cases, though, where you could say there is a need and even an ethical duty to modify human beings. And the one case is if you have two people who want to have a child, but they're both carrying, they're homozygous, there's no way they can have a healthy child, and they have a disease. But there is a way you could modify the embryo to eliminate a disease. And you tell someone, wait, listen, we can get rid of your disease, but we're just not going to do that. You have to have a diseased child. Would be unethical if the technology works and is safe. That's one case. But the second case is space. I think if you think about going to a planet where you cannot possibly survive unless you are modified to do so, barring mechanical or other ways to protect you, if they don't work, but genetics could work, then if that's the only way you can, then you have to do it. You have to enable them to survive and be safe.
So how does that constitute saving humanity? I mean, what are you saving humanity from, except, exporting people or migrating people to another planet?
Yeah, I don't want to leave this planet. I love Earth. I've been to a lot of planets. This one's my favorite so far, for sure. But Earth,
even though it's filled with problems.
It's got problems, but every planet has problems. It's fine. But I think this is, I don't think of other planets as a way to escape. I view them as a way to survive. It's not plan B, it's plan A. The reason being, most days when I wake up, the first thing I think of is the sun engulfing the Earth. In about a billion years, it'll get too hot because the oceans will boil. In about 5 billion years, the Earth will literally not exist anymore. And this is a long time away, but I think of it daily because that means, by definition, no matter how wonderful humans are, or what we create, or what we've done, if we're still here, everything will be lost except for the radio waves we transmitted into space. And I think about this every day, multiple times a day, but not with sadness, but with motivation. I think, well, that means we have a finite time on this planet as a cosmological fact. So we should get to work. We should get to work on figuring out ways we can survive on other planets, ways to cure diseases, ways to live longer, live healthier. So I view the inevitable destruction of our planet as a motivating positive factor.
So are you free this weekend? Because I'm leaving to go to Dubai for COP28, and there is a one-day focused on health. We need to bring this message to COP28 because
I have one of my companies will be there, so some of the members, but I am not currently going to be there.
Yeah. Not that we shouldn't try to solve, the climate health and save the planet and all of that, but there's an additional value add in terms of sending people to space. Yeah. So
and what we learn by pushing technology to its limits, to reducing the mass to get something to work in space, making sure it works with complicated fluidics and microgravity. It's a selection pressure on technology that makes it smaller, lighter, better, more adaptable. So spaceflight is people often say, well, why do this work in space? Because there's problems here on Earth, and I think that's a false dichotomy. You can work on cancer and you can go into space.
Do you think there's life in other planets already?
So of course, we don't know. So far, the answer is there's no evidence of it. But I think there's probably microbial life. The old famous question is the Drake equation of the estimates of how many planets could have life, or Enrico Fermi's question of where is everybody? Yeah. He famously said, if there's so many planets and life is possible, well, where is everybody?
Yeah, exactly.
And I actually think it's the wrong question. I think when is everyone else going to get here is the question. So it's 13.7 billion years of the universe has been here. The universe had to cool for several billion years. You couldn't even make planets for a while. We've had several billion years, 5-6 billion years, where planets have cooled, formed, solar systems formed. Then you had to make complex molecules, you had to make life. So I think we have only had 4 to 5, maybe 6 billion years where life even could be made. And we have trillions of years ahead of us where life will be made. So I think we might just be early. We might just be the first ones to the party, which is fun. So we get to.
So you can't save humanity alone. What does everybody else here need to do? Because you are brilliant, but still you need help from the rest of us.
Absolutely, and it's very kind of you. I think I'm just weird, and I read a lot. I don't know, but I think, in writing the book, I had this moment. Where I originally in my head, since I was a kid, I thought 5 billion years. We have 5 billion years. It's a long time. And I was finishing the book, updating parts of the last chapter, which is mostly about the end of the universe, which was kind of sad to write. But I had realized that about 1 billion years, the oceans will boil. And so even if we're still here then, we don't really have 5 billion years. We have about 1 billion. And so I was in the kitchen downstairs, and my wife was like, "What's wrong with you? What's?" And I was like, I just look so sad and dilapidated. And I said, well, I just thought we had more time. And she's kind of laughed because, well, that's still 1 billion years. It's not 5 billion years, but it's a long time. I said, no, but imagine if you thought you were going to be 100 years old and someone said, well, you're going to be dead at 20. You would be devastated. So if you view all of life like that, it is devastating. And so to answer your question, what do I want people to do? The first thing I want is everyone to have a time-independent view of the value of life, that we, current life, future life, past life, as far as we know, is only on this planet. It may be on other ones, but so far this is it. And there's only one species that has awareness of extinction, which is us. And I just want more people to be aware of their capacity to think long term, to not just, I mean, it's easy for us to say that in a nice room with cocktails and food, and people have jobs and houses, and the majority of the world is in a much worse state. But more and more people being lifted out of poverty helps us get to that goal. So there's socioeconomic benefits that help. Scientific development helps us actually survive in space, get more tools to underserved populations around the world. But I think awareness is really the first part, is that even if people don't have a lot of resources, just knowing that they have the ability to think long term and to act that way would change, I think, society for the better.
You're clearly a polymath, right? You're trying to save the world. You look at things from up above, and then you're looking things at the very microscopic level and the macroscopic level. And speaking of microscopic level, or maybe even nano level, I mean, it looks like you're like Sherlock Holmes. You're walking around swabbing, subways, cities, etc. What are you looking for? What clues? What are you investigating?
So we have a project that started mostly because my daughter, when she was very young, she licked a subway pole, as many children do. They, like many children, explore the world with their mouth. Some teenagers explore the world with their mouth, which I find disconcerting, because now I have a teenager who's 13, so I want that less of that personally. But
that's a different kind of exploration. Yes.
But as I saw that when she was a child, now as a young parent, I immediately wanted to know what has exchanged. Because every time we touch a table, we shake each other's hands. Anytime you're in a room, a crowded elevator even, we're constantly exchanging this cycle of microbes and this diurnal, continual cycle of exchange. And I just wanted to know what was there. So really it was a project born out of pure curiosity. So in 2015, it looked weird to be out swabbing a subway. People would say, what are you doing? Or what does that mean? But then during and after the pandemic, everyone's like, oh, I know what you're doing. And so it's really normalized our behavior. But since 2015, we've been out swabbing in over 60 cities every year in what's called Global City Sampling Day. It's kind of the microbial census of the planet, mostly in urban areas, to see what's there. And we're looking for new species, antibiotic resistance, and new biochemistry. Those three things help us literally discover capacity of life that we haven't known before, very analogous to what CRISPR was. CRISPR was just found by poking around just to see what's there. And along the way, we discover so many species and new functions. And so I view it as a continuing to build up the capacity of what we know about life and what it can do.
Well, there's a piece of me that's thinking, Sherlock Holmes, CSI, crime scene investigation. So you're looking at it from a positive, right? What species exist. But, during COVID, we were all swabbing in other ways, and we were, and,
hand
sanitizing, washing groceries with Clorox before we ate it, etc. Right? What are your thoughts about that?
I think, I used to have some slides in a talk I would give and say, most of the microbes are fine, it's mostly fine, but now I have a slide that says, except during pandemics. So, generally fine, but
because every so often
there will be a circulating virus, or what's extraordinary, I just had a PhD student in my lab today work on her PhD qualifying exam, and she's been characterizing viruses as it moves between mosquitoes and pigeons and humans. And you can see the functions of the virus, it changes as it moves between species. So, which we had no idea of until we've been able to see how the viruses move. So I think my solace is that there has always been an immune system battle with the environment. And if you're generally healthy, you don't have to worry too much about it. But if you're immunocompromised or really old or have other diseases, we now know that, and we can help protect you, or even, I think, maybe boost your immune system. But I think it's changed my view a bit of the biology. I used to think, well, it's this wonderful ecosystem, and we're all surrounded by microbes, and it's kind of this happy-go-lucky state. But
there are these bad apples.
there are some bad apples. Or even just if you're a long-standing HIV patient and you've lost all your T cells, look at what happens to weird skin infections, odd fungus that will grow quite literally on you because your immune system is attenuated. So we know
we're fragile.
It's this idea of it being a wonderful, oh, they're all with us and we're all microbes and we're all eukaryotic cells, prokaryotic cells, let's all hang out. It's not so much like that. It's really a consistent battle between the inside world and the outside world. And we mostly are okay. But now I take solace also in the fact that we can now see them. We can find a new virus, sequence it, develop vaccines within a matter of weeks or even days. And that's extraordinary.
Thank God.
Yeah.
Right? So we have questions from LinkedIn. This one's interesting, right? Have you considered looking into aerobic exercise as a means to optimize elastin content prior to long duration spaceflight, as elastin appears to degrade with radiation exposure, and elastin appears to degrade with aging as well? Yes.
But you're 25 or whatever it is. I have.
It's the aerobic exercise. Yeah.
I think so. So we have looked at the questions and some people online here. So yes, and actually the astronauts, we monitor their workout behaviors, their blood work, their urine, their stool, everything we can we collect from the astronauts. In fact, we collect wastewater also as part of monitoring. Every time someone flushes a toilet, and if no one takes a sample, I die a little bit inside. Because every flush has a lot of information that if I can, we'd like to capture that. But if for elastin and spaceflight, they have to work out usually at least once a day, sometimes twice a day for most of the crews, just to avoid the loss of merely bone density, the muscle tone. And it is pretty aerobic, but a lot of it's strength workouts. So I think that is a great question because we need probably multiple kinds of workouts that are not just strength building to, I think, mitigate that damage of both radiation and weightlessness.
Let's see if the audience has any questions. Yeah. Yes.
This is a great talk so far. I would say my question is, you said that you have been collecting a lot of samples with swabs. Is there a way that I could get this data?
A good question. So all the swabs collected and sequenced are online when we publish the papers, but also we have a new platform from one of my companies. It's called Biosha, which creates this platform for taking any piece of DNA that's been sequenced to actually type it in. You can see where it is across the world and monitor it. So you can go to GeoSeek is another website you can go to, but also anything that's public is usually coming from papers, and we try to make every data public that we can.
Another question.
So I hope this is not the last question because it's a pretty gloomy one.
Ooh.
But,
Yes.
I can try and turn it around. Maybe.
Yeah.
or another question. But you look at what's happening on Earth, and you have to wonder, is the reason we haven't seen any other intelligent life because any sufficiently intelligent life is intelligent enough to destroy itself kind of inadvertently because it becomes inevitably short-term and money-driven, and just the bad guys get hold of the powerful stuff.
So yeah, the self-destruction question of where is everybody, which is one of the options that Fermi discussed and many people have thought about since. So it's definitely it's a possible reason why we haven't heard from anybody out there. But I think the only data set we have is our own in terms of a allegedly intelligent species that's trying to work with each other. And I think there's plenty to make you sad based on just, people can be selfish, they can be cruel, they can, my daughter gets mean text messages from her 13-year-old boyfriend that are totally unnecessary. And, but, some of it is youth where people are young. And I think as a species, we are still relatively young. But the upside is, and actually I think the context that's really needed is, the last few thousand years, it was really hard to do anything good on a species level. And most importantly, it was actually impossible to do until really the last few decades. There was no means by which to coordinate on a global scale to address any global problem until the 60s, 70s. You could do telegrams, you could send letters, but it was barbaric and it was tribal because that was the only way you could survive. But what's happened is we now have, for example, a hole in the ozone layer, and there was both an economic driver as well as a social driver for people to say, well, there's a global problem, let's solve it as a global community, and we did, at least once. And so far, only really once, the ozone layer, but it means it's possible. And it hasn't quite happened with climate change, but the awareness is better today than I think it's ever been. And we're looking at all means by which to attack it. And I think I think there is at least one example. The other thing is just people sharing more than ever before in terms of data, algorithms, tools. The diagnostics have gotten better. Actually, there's another company, Nurture Genomics, that's here tonight. Hans is the CEO, where you can sequence at birth, and you can avoid diseases that you would have had no idea were there even 5 years ago. And so I think there's way, and I know you've been an early investor in 23andMe, for example, and big advocate, actually one of the first of the personalized genome project. So there is, more people are giving their data to the world than ever before. So I think there's plenty of signs of hope. And if some of the basic metrics, look at infant mortality, literacy, overall life expectancy, all those things are better than they've ever been, except they dipped on a little bit during COVID, and fentanyl. So it's overall good, but it comes down again. But I think it's 2 steps forward, 1 step back. But overall, the progression is there. And we're a young species, so I think we have to give ourselves a little credit that we've made it this far, and that we can now. You can call anyone in China or India, or you can call someone in Sweden, and none of us think twice about it. It's actually a global community, but we're only. Imagine if we're a 20-year-old kid. That's basically what we are as a global community. So it's kind of a miracle we solved, I think, the ozone hole problem as early as we did. So now we just have to think about information content. Not getting trapped in misinformation. We have new responsibilities with the global community that we've never had to think about.
Can you genetically modify
Could you make people kinder?
Kinder,
could
gentler?
you make them less kinder, gentler machine gun man?
Yes,
Could you? So this actually comes up also with some of the questions about de-extinction. As you're starting to create new species or bring them back, well, do you bring back the whole species or do you modify a little bit? Or
what about Winnie the Pooh?
Winnie the Pooh, something really nice. I like honey. Maybe we could just dip our whole body in honey.
Yes.
It's a bathtub full of honey,
Yes.
and then have barbecue sauce on top of it. That sounds great to me. I think I would
maybe
love
not
that.
Eeyore,
Yeah,
but maybe Winnie.
But I would. There is some of this happens through selection. Of course, there's a fight or flight response that's been selected for millions of years. That's still embedded in us, and I think some of it even just learning how the brain works, learning how to control your thoughts, even with meditation. There's practices that we have today that we didn't have 10,000 years ago that are helpful all by themselves. And so you might not need to genetically engineer people to be nicer. We could maybe learn how to be nicer. But there is, some of the pharmacological interventions are trying to do just that, trying to make people calmer, remove depression. They're a small molecule version of fixing something that you could potentially do genetically. It's too early to tell if that would be good, but you can imagine if you send a bunch of people to Mars and you start a whole new civilization, there's going to be a founder effect where whoever those first few hundred people are will dramatically determine what everyone looks like on that planet. So I think we'll come to see that pass.
One LinkedIn question, and we'll keep an eye on time. What do you think is the biggest health physiological barrier for human missions to Mars?
So there's the biggest risk to being on Mars is the radiation is one we think a lot about, just because, especially the way to get there. And once you get on the planet, you have a whole planet beneath you that's defending you against any incoming radiation, and you can go underground. But on the way there, especially, and even on Mars, there's no magnetosphere. There's 1% atmosphere. There are new toxins in the soil, perchlorates. It will be a toxin-rich and radiation-heavy environment, and it will be hard. So I think you could. One thing we're doing in lab is actually turning on genes for radiation repair earlier, taking them from tardigrades. So you can actually control genes much like you would a light switch and turn them on where you need them. So we could maybe do this for the Martians that would be there. I think the psychosocial ones are also a big challenge. If people, have you ever been in a car with someone for like 10 hours and after a while they start farting in the car, they're making weird noises, they're chewing gum too loud, they're talking, they're on the phone, things can happen. And that's just in a car, right? So I'm not saying I do any of those things, but I've seen this happen with others. But and if you're on a trip that's 6 to 7 months, just a one-way trip, and you get there, and there's no more than 20 people around you for at least another year, it was stressful. So the psychosocial stress alone is something that is a big factor. And
or you could be like Matt Damon in The Martian.
that's right, and have to
and be
grow
stuck there.
grow
Yeah,
potatoes
and
from your own stool.
Grow potatoes.
Which had a flavor I'm sure we didn't hear
Yeah,
about in
with-
the book.
yes.
But the,
Tastiest potatoes ever.
that's right, it kept them alive. So I'm sure he was happy. And it's a great book, by the way, I read the book. Also the Hail
We
Mary
should make a
Project.
movie where instead of him growing potatoes, somebody, is genetically modifying him so that he can adapt.
So this is one thing I wrote a bit about in my recent book is I think it is really infuriating that we have essential amino acids. There's
Yeah.
9 essential amino. Why can't we make all of our own amino acids?
Yeah.
That is just bullshit. Is the only way
Yeah.
to call it. It doesn't make any sense. So we could take a few genes from other species, complete the circuits in our own cells, so we can make all of our own amino acids, or even vitamin C, for example. We used to make our own vitamin C, but now you have to get it from your diet. If you don't get enough, you'll get scurvy. It's very sad. But a lot of wet-nosed primates make their own vitamin C, what's called a gene of gulo. Dogs, cats, they all make their own vitamin C. They don't need mojitos to get their vitamin C. But we need to get it from the environment, mostly because nutrition has reduced that selection pressure. But the gene is still inside of us. We could modify it, turn it back on. So I love these ideas of taking genes inside of us that are still there and just turning them back on. Or the cures for beta thalassemia or sickle cell disease are turning back on fetal hemoglobin inside of you. The gene is inside all of us still today. And if you have a problem with your adult hemoglobin, well, let's turn back on the gene you had when you were a fetus to cure the disease, which is phenomenal, right? And that's, it just was approved for the UK. It's likely getting approved by the FDA, I would guess, within 6 months. And so this
Wow.
concept of turning back on
Yeah.
genes inside of you as a cure is happening. And so it's phenomenal.
Yeah, so, speaking of, science fiction actually becoming fact, that's what's happening in the world, and it's possible. And I could sit here and talk all day long, all evening long, I should say, but I know we have to wrap. But thank you for joining us. Thank you for a really exciting conversation. And here's to the next 500 years.
Thanks. Cheers. Thank you very much.
Cheers.
Pleasure.
Cheers, everybody.